Hey everyone, my name is Naisargi. Welcome back to my channel. In today's video, I am going to share with you all the first and foremost basic concept which you need to learn to work in a biology or a chemistry laboratory or any laboratory per se. The good laboratory practices. Now, I'm sure many of you might be aware of this concept. Some of you might also be unaware of this concept, and some of you might also be aware but still neglecting these things while working in the lab. When you perform an experiment in your laboratory, you should be able to reproduce those experiments again, and you should also be able to upscale those experiments. That means that the integrity of your work should be maintained. The proper conduct while working in the lab should be maintained in order to ensure the quality of your work. When you are working in a laboratory, there are certain basic protocols that you need to keep in mind to ensure your own safety as well as to ensure the safety of the environment. So, these protocols by which you abide while working in the lab, these are known as good laboratory practices. So, one by one, we will be going through each of these good laboratory practices, or GLPs as we say. We will learn the whats and the whys of each of them. First and the foremost thing, wear protective gear while working in the lab. You have to ensure that you wear a proper full-sleeved apron. Your eyes should be covered by protective glasses. Your feet should be covered by proper shoes. Now, I cannot stress the fact enough that you need the protective gears while working in the lab. I'll share with you one incident that occurred with me. While I was doing my PhD, there was a junior of mine who was doing her masters, and they were supposed to do the simple experiment of acid and base neutralization in the laboratory. For that, she was carrying a beaker full of H2SO4 with her, which she accidentally spilled on her foot, and she had a severe burn. She had to be rushed to the hospital. So these mishaps do occur, so please make sure that you protect your feet, even your hands, by using proper gloves. When you are working in the lab, you come across various chemicals which can be carcinogenic or simply they burn your skin. You do not know which chemical can cause what harm. In case you're doing an experiment which is related to the handling of a microbes or any other cell line or isolation of DNA, suppose, this will also ensure that the microbes or the DNA from your hands and your breathing do not enter your workspace. Here I would like to make a special mention to those people who wear their lab coats on vehicles or to the restrooms or to the college canteens. Guys, please do not do that. It just ruins the whole purpose, because in these environments like your restroom or your canteen or on your vehicle, there are n number of germs. They get stuck on your lab coats, and ultimately when you work with the same lab coats in your laboratory, there is a heavy chance that these microbes get mixed with your working environment and they can cause various contaminations. In addition, if in between your experiments you need to open or close the doors or any lockers, etc., please remove your glove of that hand. Use the bare hand to handle the door knobs or locker knobs, and after you're done, wear your glove again on that hand. This will ensure that your glove does not touch any other surface apart from your working environment. In addition, the lab coats that you wear should be separate for every lab. For example, if you're working in an animal house facility, in the cell culture facility, in the microbial culture facility, or in your normal routine lab, for each of these labs you should keep separate aprons. Ideally, you should autoclave your lab coats, but it's okay if you even wash your aprons thoroughly using a proper detergent and warm water. You have to discard your gloves after every use. In addition, you have to make sure that the gloves that you're wearing are of appropriate size of your hand. If your gloves are too loose, then your grip to hold the experimental animals or burettes or pipettes will be compromised. If you have to give, suppose, injection, or you have to incorporate the dose in your experimental animals, or you have to handle the cell lines or anything. If your gloves are too loose or too tight, it won't be comfortable for you to work, and it may also lead to the causing of errors. Secondly, again a very small point but a very significant one, make sure you label each and everything in your laboratory. Your beakers, your tubes, your Eppendorfs, be it anything, you should have a proper labeling on everything. Suppose you are doing 2 or 3 things simultaneously, or if you are in research, you are simultaneously analyzing your data, doing some calculations and doing your experiment, there's quite a possibility that you will be making errors while mixing the solutions if you do not know what solution is there in which beaker or which tube. For the purpose of labeling, you have to use a proper marker which is waterproof, or you can use labels, and after you put your label or the marker on your glassware or your plasticware, make sure to cover that labeling with a proper transparent sellotape. This will ensure that your labeling does not go off even if any chemical or water touches it. For labeling your cryo vials, you have specific cryo tags available, because those tags, they do not come off even when you store your vials in as low temperatures as -80 degrees Celsius or even in the liquid nitrogen. Which brings me to my next point of discussion, which is the proper storage of your working chemicals. Now, whenever you order any kit or any chemical from the market or from any vendor, on the box or the bottle of that chemical, its storage temperature is written. So you have to make sure that you are storing your chemicals at appropriate temperature so that they do not go bad till its expiry date. The next point is the positioning of your working desk. It is very important that you position your working desk accordingly so that you are the most comfortable and relaxed while you're doing your experiment. This will ensure that you cause as minimum manual errors as possible, because your mind will be at ease while you're performing your experiment. So what does this mean? For example, I am a right-handed person. When I am working in an animal cell culture facility, I make sure that I keep my micropipettes, pipette tips, as well as the media bottles on my right side, while I put my discard beakers on the left side, because I do not want to put my discard beakers and the media bottles on the same side. So this will ensure the ease of my working. I'll use my right hand to pick up the pipette, the media, and I'll put it in my flask or remove it or whatever, and then I'll just gently discard the media in the discard beaker on my left hand side. Which brings to my next point, that you always have to keep your discard beakers, your markers and your alcohol and tissue paper ready whenever you're doing any of your experiment. Make sure that you keep your solid and liquid waste discard beakers separated, because the disposal of both kinds of waste is different. I am going to make a whole separate video on biohazardous waste disposal protocols. You have to wipe clean the surface of your working environment using a tissue paper and alcohol before as well as after you work. You have to ensure that your working space is dust free, it's clutter free, as well as it is organized. This will ensure that your mind is also clutter free, it is organized and at comfort to work optimally in your working environment. The next thing, point towards your own personal hygiene while working in the laboratory. You have to make sure you wash your hands thoroughly till elbow before and after you are done working using warm water and soap solution. Secondly, you have to tie your hair and cut your nails while working in the laboratory. Suppose you are working with the burners. You do not want your hair to come in contact with the flame or the chemical and ultimately cause any mishap, right? Also, the loose hair entraps a lot of microbes, and you do not want your experiment to have any contamination because you work so hard with it, right? The third thing, you cannot use your cell phones while you're working. You cannot have any food or drink in the working environment. This will ensure your own safety as well as the integrity of your experiments. The next point ensures the validity of your experiments and its data. So for that, you have to repeat any experiment at least thrice at different time points, and each time that you do the experiment you have to keep at least triplicates. So this will ensure the accuracy as well as precision and the reproducibility of your experiments, and this way you will be able to generate the statistical significance in your data. The next point, again a very important one, always put acid in water. You cannot put water in acid or else it will just go boom. There is this concept of free energy. When water reacts with acid, it's an exothermic reaction. So if the quantity of acid is more than the quantity of water, then it will cause an explosion. But if you put acid in water drop by drop, however less or more dilution you want to make, slow free energy will be released, and that is why there won't be any explosion. Last point, but definitely not the least, you have to maintain your daily record book while you work in your lab. I cannot stress this fact more, because a lot of people, they fail to maintain a proper record book when they work in the lab, which is not a good practice. Trust me guys, because this might seem small, but actually when you are dealing with a lot of things together, suppose if you are in a graduate level college or in high school, you might have to attend so many lectures, so many practicals, so many tuition classes. You have everything in here which goes away after a certain point of time. You need to write it down, however small it may seem. If you are in a research facility, you are dealing with so many tasks at a given point of time. You are doing data analysis, you are doing 2-3 experiments together. There are high chances that you forget stuff or you make manual errors. For that purpose, you need to write down each and everything that you did, that you read, that you recorded, everything. Even when you go to your teacher or your supervisor for the discussion of your experimental data, you have to be very clear and very precise with what you did. In addition, if you have written down everything precisely, it will help you reproduce your experiments the next time, or even in the troubleshooting if you did not get your results in the first go. So this was all for today's video, you guys. Please stay safe while working in the laboratory. Please ensure the quality of your data, and please ensure the safety of the environment as well. I'll soon come up with my next video. Till then, you guys keep spreading kindness and keep spreading knowledge. I'll see you all soon. Bye bye.